Hello students, today I bring for you a new chapter, that is biomolecules. We are going to cover all the topics that are in your recent syllabus. Okay, so let's start. Biomolecules are chemical compounds found in living organisms. They include organic and inorganic compounds. Analysis of chemical composition in a tissue. For that, it is a practical of class 12th you are going to do in your biology lab. So, how to bring out the chemical composition from a tissue? First of all, grind a living tissue, vegetable or a piece of liver, etc. in trichloroacetic acid to get a thick slurry. Strain this through a cheesecloth or cotton to get two fractions such as filtrate, which is acid soluble pool, and the retentate, acid insoluble fraction. Filtrate is also called acid soluble pool. It contains biomicromolecules where the molecular weight is less than 1000 Dalton. Retentate is the acid insoluble fraction, contains biomacromolecules, that is molecular weight is greater than 1000 Dalton. Next, analysis of inorganic compounds. Weigh a living tissue, weight and it is dry with to evaporate water. It is fully burnt to oxidize all carbon compounds to gaseous form CO2 and water vapor, and it forms ash. The ash contains inorganic elements and as well as inorganic compounds. Comparison. This table also in your NCERT you can see hydrogen, which is an element, which is a percentage of earth crust and human body. As you can see, 0.14 in earth crust, while it is found in 0.5% in human body. Carbon, 0.03 in earth crust, 18.5 in human body. Oxygen also here, nitrogen, sulfur, sodium, calcium, magnesium, silicon. First of all, we will discuss about biomicromolecules. Biomicromolecules includes amino acids, lipids, sugars and nitrogen bases. Molecular weight of biomicromolecule found in the acid soluble pool ranges from 18 to 800 Dalton. The acid soluble pool represents the cytoplasmic composition. First of all, amino acids. They are the compounds formed of amino group and acid group, H and a variable group R. Here you can see amino group and acid group are attached to the same carbon atom, that is called alpha carbon. So they are called alpha amino acids. As you can see in this diagram, they are substitute methanes. Here some other amino acids, R double bond H in glycine, R double bond CH3 in alanine and R double bond CH2OH in serine. The diagrams are also in your NCERT. 20 types of amino acids are used in protein synthesis. Amino acids are 3 types. Acidic amino acid, example glutamic acid. As you can see, basic amino acid, example lysine, and neutral amino acid, example valine. Some amino acids are aromatic, example tyrosine, phenylalanine and tryptophan. Amino acids can be classified as follows: essential and non-essential amino acids. Essential, first point, they cannot be synthesized by the body and should be supplied through diet. Example lysine, leucine, isoleucine, tryptophan, etc. Non-essential amino acids they can be synthesized by the body, example glycine, alanine, serine and arginine, etc. In amino acids, NH2 and COOH have ionizable nature. So structure of amino acids changes in solutions of different pH. If both NH2 and COOH are ionized, it is called deuterion. As you can see in this diagram, deuterionic form. Now coming to lipids, these are water insoluble, contains carbon, hydrogen and oxygen, but number of oxygen atom is less. Types of lipids, simple lipid, compound and derivative lipids. First of all, simple lipids. These are formed of fatty acid and alcohol like glycerol. Fatty acids are lipids with a hydrocarbon chain R group ending in COOH group, that is R bond COOH. Example, palmitic acid has 16 carbon. Here the diagram also given. You can see CH3 CH2 whole 14 bond COOH or C15 H31 bond COOH, and arachidonic acid has 20 carbon. Types of fatty acids. Saturated fatty acids, they have no double or triple bond between carbon atoms. Example, palmitic acid, stearic acid. Unsaturated fatty acids, they have one or more carbon bonds. Example, oleic acid and arachidonic acid. Structure of glycerol, trihydroxypropane. Here you can see fatty acids are esterified with glycerol through ester bond forming monoglycerides, diglycerides and triglycerides. As you can see how a fatty acid or a palmitic acid makes a bond with glycerol through a ester bond. Here you can see 1 glycerol plus 1 fatty acid equal to monoglycerides. See the diagram. 1 glycerol plus 2 fatty acids, diglycerides, and 1 glycerol plus 3 fatty acids, triglycerides. Triglycerides, here R1, R2 and R3 are fatty acids. As you can see in this diagram, based on melting point, lipids, triglycerides are two types: fats, which have higher melting point, and oils that have low melting point. Next is your compound lipids. These are the esters of fatty acids and alcohol with additional group. Example, phospholipids, fatty acids plus glycerol plus phosphate. They are found in cell membrane. Example, lecithin. As you can see in this diagram, where you are aware about cell membrane, the structure of cell membrane, here you can find out the lipid bilayer. So it is what? It is a phospholipids, which is made up of fatty acids, glycerol and phosphate. Derived lipids, they are the product of hydrolysis of simple lipids and compound lipids. Example, cholesterol. Third is your sugar, carbohydrate. Sugar are sweet and water-soluble carbohydrates. They are formed of C, H and O in the ratio of 1:2:1. Example, glucose, ribose, etc. Glucose C6H12O6, ribose C5H10O5. Next is your nitrogen bases. These are the nitrogen containing cyclic compounds found in nucleic acids. They are classified as follows. Nitrogen bases divided into two parts: purines, pyrimidines. Purines are adenine, symbolize A, guanine, symbolize capital G, and pyrimidines are three types: cytosine, C, thiamine, T, uracil, U. Nucleosides. These are the compounds formed by link of a nitrogen base with a sugar. When a nitrogen base linked with a sugar, then it forms nucleoside. Adenine plus sugar, you can see adenosine. Guanine plus sugar, guanosine. Cytosine plus sugar, cytidine. Thiamine plus sugar, thymidine, and uracil plus sugar, uridine. See the diagram, you can easily find out the nucleosides. And what are nucleotides? Now these are the compounds formed by linking of a nucleoside, which is also made by nitrogenous base and a sugar with a phosphate. So nitrogen base plus sugar plus phosphate gives rise to nucleotide. Adenine plus sugar plus phosphate gives rise to adenylic acid. Guanine plus sugar plus phosphate gives rise to guanylic acid. Cytosine plus sugar plus phosphate equal to cytidilic acid. Thiamine plus sugar plus phosphate equal to thymidilic acid. Uracil plus sugar plus phosphate equal to uridylic acid. Here is the example of adenylate acid. As you can see, a nitrogen base or adenine plus sugar plus phosphate is also added. So it forms a nucleotide. Nucleotides are heterocyclic compounds. Heterocyclic compounds. Nucleic acids, which is made up of DNA and RNA, are made up of nucleotides. So the DNA is made up of this type of nucleotides. Okay, DNA and RNA both. Next, biomacromolecules. These are biomolecules having molecular weight greater than 1000 Dalton. They include proteins, polysaccharides, nucleic acids, etc. Their molecular weight is 10,000 Dalton and above. Acid insoluble fraction. Macromolecular fraction includes macromolecules from cytoplasm and organelles. Structure of cell membrane, as you can see, lipid is not strictly a macromolecule as its molecular weight does not exceed 800 Dalton, but it comes under acid insoluble fraction because many lipids are arranged in structures like cell membrane. When a tissue is grinded, cell membrane are broken and forms water insoluble vesicles. They cannot be filtered along acid soluble fraction. So that's why lipid is not strictly a macromolecule. Average composition of cell: water 70 to 90%, protein 10 to 15%, carbohydrate 3%, nucleic acids 5 to 7%, and ions are 1%. First of all, proteins. Proteins are heteropolymer of amino acids. They are polypeptides, that is, linear chain of amino acids linked by peptide bond. Peptide bond is formed when COOH group of one amino acid react with NH2 group of next amino acid by releasing a molecule of water, that is called dehydration. As you can see, chain of amino acids and how the peptide bond is arranged between these amino acids. Proteins, functions of proteins: for growth of tissue repair, transport nutrients across the cell membrane, example GLUT4 enables glucose transport into cell, act as intercellular ground substance, example collagen, act as antibodies to fight infectious organisms, act as receptors, example receptors of smell, taste and hormones. Some are hormones, example insulin, some enzymes, example trypsin, some are pigments, that are hemoglobins, etc. So protein, most abundant protein in animal world is collagen. Most abundant protein in biosphere is the rubisco. Full form: ribulose, bisphosphate, carboxylase, oxygenase. Structural level of protein: primary, secondary, tertiary, quaternary. There are 4 different type of structural levels of proteins. Let's see. You can see the diagram. Primary structure. It describes the sequence of amino acids, that is positional information in a protein. Left end, first amino acid, N terminal amino acid. Right end, last amino acid, that is called C terminal amino acids. And between them peptide bond is there, as you can see in this diagram. Secondary structure: here one or more polypeptide chains are folded in the form of a helix. It has only right hand helix, example keratin, fibroin, which is a silk fiber. Next, some secondary structure: fibroin, silk protein, antiparallel beta sheet rich in alanine and glycerin, small side chain allow closed packing. As you can see in this diagram, here it is a keratin, typical strand of hair. So these are the secondary structures protein. Now tertiary structure. As you can see, tertiary structure: alpha helix, beta strand, and they both form tertiary structure. Myoglobin is one of an example. Here helical polypeptide chain is further folded like a hollow woolen ball. It gives 3D view. Tertiary structure is necessary for many biological activities of proteins, example myoglobin, enzymes, etc. Next is the quaternary structure. Tertiary structure forms quaternary structure. As you can see here, more than one polypeptide chain forms tertiary structure, and each chain functions as a subunit of protein. Example, hemoglobin. It has 4 subunits: alpha, 2 alpha subunit and 2 beta subunits. Now, proteins at a glance. Primary, as you can see, amino acids, beta plated sheet, alpha helix, polypeptide chain, assembled subunit. Primary structure, secondary structure, tertiary structure, quaternary structure. Second is our polysaccharides. These are polymers of sugar. Monosaccharides. Example, starch, homopolymer of glucose, cellulose, homopolymer of glucose, glycogen, homopolymer of glucose, insulin, homopolymer of fructose. Here, there are complex polysaccharides formed of amino sugar. Example, glucosamine, N-acetyl, galactosamine, etc. Chitin is the homopolymer of N-acetyl glucosamine. It is seen in exoskeleton of arthropods and fungal cell wall. Next, glycosidic bond in polysaccharides. As you can see, it is a bond formed when individual monosaccharide are linked between 2 carbon atoms by dehydration, and they form glycosidic bond. Okay, see the linkage. 2 monosaccharide subunits, they attached with a glycosidic linkage, and water is lost. Diagram representation of portion of glycogen. Here the diagram also given in your NCERT. Next, polysaccharides. Starch forms helical secondary structure. It can hold I2 molecule into the helical portion, giving blue color. Cellulose has no complex helix and therefore cannot hold iodine. Third, nucleic acids, DNA and RNA. Nucleic acids are heteropolymer of nucleotides, that is, many nucleotides are linked to form polynucleotide. So you can see a nucleotide, a nitrogen base, sugar and a phosphate molecule is attached. Nucleic acids, DNA and RNA. Nucleic acids are 2 types, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Here you can see secondary structure of DNA Watson-Crick double helix model. There are more than a dozen forms of DNA such as A, B, C, D, E, Z, etc. DNA consists of 2 polynucleotide strands arranged antiparallelly as a double helix. See these diagrams? A major groove, minor groove, and 3' to 5' end, 5' to 3' end, and between them how the nitrogenous bases are attached. And you can see both the strands equally make a distance of 2 nanometer, and both the nitrogenous bases have a gap of 0.34 nanometer, and you can see a complete helix makes the distance of 3.4 nanometer. Sugar phosphate backbone, nitrogenous bases, major groove, minor groove. Here again the same structure, Watson-Crick model. In DNA, a nucleotide consists of nitrogenous base, deoxyribose sugar and phosphate molecule. See this diagram? Nitrogenous bases are there, pentose sugar, also called deoxyribose sugar, and a phosphate group is attached. Backbone or strands of DNA is formed by the sugar phosphate sugar chain. Steps are formed of nitrogenous base pairs. Nitrogenous base includes adenine, guanine, thymine and cytosine. Uracil is absent. Again, adenine pair with thymine by 2 hydrogen bond, guanine pair with cytosine by 3 hydrogen bonds. See the diagram. You can get a clear view how the bonds, hydrogen bonds formed between the nitrogenous bases. A phosphate molecule linked the 3 carbon of sugar of one nucleotide to the 5 carbon of the sugar of the next nucleotide. As you can see, the phosphate molecules are also attached. Bond between phosphate and OH group of sugar is an ester bond. We already comes with this contact, ester bond, with this term. As there is one such ester bond on either side, it is called phosphodiester bond. So both sides there are two ester bonds are there. That's why it is called phosphodiester bond. The bond between sugar and nitrogen base is called N-glycosidic bond. The bond between sugar and nitrogen base is called N-glycosidic bond. Remember the term. In bDNA, one full turn has 10 steps. As you can see, 10 base pairs. Length of one full turn is 34 angstrom. That is 3.4 angstrom for each step. At each step, the strand turns 36 degree or 360 degree for a full turn. Next is your enzymes. Enzymes are biological catalysts which influence the speed of biochemical reactions. All enzymes are proteins, but all proteins are not enzymes. Enzymes are specific, that is, each enzyme has its own substrate. Ribozyme, nucleic acids, RNA that behave like enzyme. Enzyme form tertiary structure with some surface pockets called active site into which the substrate fits. Chemical reactions. Chemical compounds undergo two types of changes. A physical change, that is a change in shape or state of matter without breaking bond. Example, ice melt into water and water becomes vapor. Second, chemical change or chemical reaction. In this, bonds are broken and new bonds are formed. It may be organic or inorganic reaction. Example, in inorganic, BaOH2 plus H2SO4 gives rise to BaSO4 plus 2H2O. Organic, starch plus H2O gives rise to glucose. Here you can see heat energy in, bonds breaking, heat energy out, bonds making. Okay. Rate of physical or chemical process equal to amount of product formed per unit time. Rate of velocity if the direction is specified. Rate of physical and chemical processes are influenced by factors such as temperature. Generally, rate doubles or decreases by half for every 10 degree change in 10 degree Celsius change in either direction. Rate of enzyme catalyzed reactions is very high. Example, carbonic anhydrase is the fastest enzyme. It accelerates the following reactions 10 million times. In the absence of the enzyme, only 200 molecules of H2CO3 are formed in an hour. If the enzyme present, that is the carbonic anhydrase is present, about 6 lakh molecules are formed per second. So you can see how fast the enzyme make a reaction very high. So it is shown 10 million times. Next, in a metabolic pathway, each step is catalyzed by different enzymes. Example, in glycolysis, 10 different enzymes takes place. Glycolysis means the formation of glucose to pyruvic acid. So there are 10 different enzymes takes place. As you can see in the first step, hexokinase. Second step, phospho-glucose isomerase. Third, phosphofructokinase. Fourth, aldolase. Fifth, triose phosphate isomerase. Sixth, you can see glyceraldehyde phosphate dehydrogenase. Next, phosphoglycerate kinase, phosphoglycerate mutase, enolase, and then finally pyruvate kinase. So this particular glycolysis step having 10 different enzymes. Okay. Next, enzymes act with substrate like a lock and key model action. It includes the following step. Substrate binds to the active site of enzyme. You can see the diagram, E plus S, the E for enzyme and S for substrate. This induces some changes in enzyme so that the substrate tightly bound with the active site of the enzyme to form enzyme substrate complex. ES is formed. The active site breaks chemical bond of a substrate to form enzyme product complex EP, and then you can see the enzyme released the product and the free enzyme is ready to bind to the other molecule of the substrate E plus P, last one. This pathway goes through some unstable transition state structure. Concept of activation energy. How do enzymes speed up a chemical reaction? You can see activation energy is the additional energy required to start a chemical reaction. In an exothermic or endothermic reaction, the substrate must go through a much higher energy state. It is called transition state energy. Therefore, activation of energy is the difference between average energy of substrate and transition state energy. If the product P, see the diagram, if the product P is at a lower energy level than the substrate S, the reaction is exothermic reaction, that is spontaneous reaction. It requires no energy to form the product. In a biochemical reaction, enzymes lower the activation energy. As a result, speed of the reaction increases. Factors affecting enzymatic activity: temperature and pH, concentration of the substrate, and the presence of inhibitor. First of all, temperature and pH. Enzymes show highest activity at optimum temperature and pH. Activity decline below and above optimum value. At low temperature, enzymes temporarily inactive. At high temperature, enzymes destroyed because proteins are denatured by heat. Inorganic catalysts work at high temperature and pressure, but enzymes get damaged at high temperature, so they have to work below 40 degrees Celsius. Thermophilic organisms have enzymes which are stable at high temperature up to 80 to 90 degrees Celsius. Concentration of a substrate. When the increase in substrate concentration, the velocity of enzyme act rises at fast and reaches a maximum velocity, Vmax, you can see. This is not exceeded by further rise in concentration because enzyme molecules are fewer than the substrate molecules. That is, no free enzyme molecule to bind with additional substrate molecules. Presence of inhibitor. The binding of specific chemicals, inhibitors, shut up the enzyme activity. This is called inhibition. The inhibitor, closely similar to the substrate, is called competitive inhibitor. It competes with the substrate for the binding site of enzymes. As a result, the substrate cannot bind and the enzyme action declines. You can see this diagram. No competitive inhibition, and here the competitive inhibition is occurred. Example of competitive inhibition. Malonate is similar to the substrate succinate, so it inhibits succinic dehydrogenase in the following reaction. Succinate, presence of succinic dehydrogenase becomes fumarate. Competitive inhibitors are used to control bacterial pathogens. Here you can see classification and nomenclature of enzymes. Oxidoreductase or dehydrogenase, S reduced to S oxidized, S oxidized, S reduced. Transferase, hydrolase, lyase, isomerase, ligase. Here you can see oxidoreductase or dehydrogenase catalyze oxidoreduction between the two substrates. Transferase catalyze a transfer of a group other than hydrogen. Hydrolase catalyze hydrolysis of ester, ether, peptide, glycosidic, C bond C, C bond halide or PN bonds. Lyase, it catalyzes removal of group by mechanism other than hydrolyze, leaving double bonds. Isomerase, it catalyzes interconversion of optical, geometric or positional isomers. Ligase, it catalyzes the linking of two compounds together. Example, enzymes which catalyze joining of bonds like C bond O, C bond S, C bond N and P bond O, etc. And finally, cofactors. These are non-protein constituents bound to the enzyme to make the enzyme catalytically active. Apoenzyme, protein portion of the enzyme, you can see apoenzyme is the protein portion and a cofactor, it becomes holoenzyme. Apoenzyme is an inactive protein but holoenzyme is the active one. When the cofactor is removed from the enzyme, its catalytic activity is lost and becomes inactive. Types of cofactor: prosthetic group, coenzymes, metal ions. Prosthetic group, organic tightly bound to apoenzyme, example heme in peroxidase and catalase. They catalyze breakdown of H2O2 to water and O2. Second coenzymes, you can see organic transient binding to apoenzyme. Many coenzymes contains vitamins, example NAD plus NADP contains niacin. Metal ions, they form coordination bonds with side chain at active site and one or more coordination bond with the substrate. Example, Zn is a cofactor of carboxypeptidase. And with this chapter biomolecules is completed. And like this video, share with your friends, and do subscribe my channel if you are new to this. Thank you. Jai Jagannath.